Let's begin with biochemistry genetics. So in genetics of biochemistry, you need to see first gene expression modification. So in the meaning itself, you can see gene expression modification. We want to modify the expression of gene. So for this, we do many experiments, especially we take mouse as our experimental tool. So in that mouse, we can either add the genes whichever we want, or we can also remove the genes. So adding of the genes is called knock in, inserting a gene. Knock out is removing a gene. So why we insert or delete the gene is to see whether how the gene expression is going to vary. So the first thing we do is we can randomly insert any kind of gene into the mouse, and we can see the constitutive expression, that is a random expression. Whereas you can also insert or delete a targeted type of gene in the mouse, so that you can get a planned, or you can say conditional expression for the targeted insertion. So this is about gene expression modification. Now coming to RNA interference, this is also a part of it. RNA interference is nothing but we are adding a small part, or can say small chip kind of RNA into our normal existing sequence. So by adding this RNA interference, we are going to inhibit the gene expression. It is mainly used for inhibition of gene expression. So here you have two types of RNA interference. One is microRNA and one is small interfering RNA. MicroRNA is a very small part. Actually, it is naturally produced in our cell itself. It is a hairpin like structure. You can see here hairpin like natural structure. By inserting this microRNA to our original mRNA, what happens is it inhibits the mRNA translation. You know, if an mRNA can't translate, then the protein can't form and the gene expression is inhibited. So this is how we inhibit the gene expression, by using a microRNA which is naturally formed in our body. So this is useful sometimes, but if this microRNA undergoes abnormal changes, then sometimes it may also inhibit a suppression gene. You know, tumor suppression gene is used for suppressing the tumors, but if this microRNA goes and inhibit the tumor suppressor gene, then the malignancy chance will rise in that particular individual. So this is about microRNA. Now coming to small interfering RNA, this is also same like microRNA, but this is somewhat an I can say exogenous product. It is produced by double-stranded RNA source from virus, and we now insert this small interfering RNA into the our normal mRNA, and the same way this also inhibits the translation. Actually, this will degrade or break down the mRNA even before undergoing translation process. This will direct microRNA will directly inhibit the translation process, whereas small interfering RNA will break down the mRNA and it will not allow to undergo into translation process. So this product, this small interfering RNA, can be chemically also synthesized. Especially we use this kind of small interfering RNA for gene knockdown experiments. That is, we ourselves wantedly want to inhibit the mRNA sequence. Then we use this. Now coming to biochemistry genetics, some genetic terms. Before that, we need to know actually this basic thing, that is gene. In the gene you have DNA, and this gene, another form, it can be also called as allele. And allele location can be understood by means of locus, and the locus is present on a chromosome, locus on chromosome. So like that, DNA, gene, allele, locus, chromosome. So now coming to genetic terms, the first one you have is codominance. It is where both alleles contribute to phenotype of heterozygote. To understand this, you need to understand what is genotype and phenotype. For example, take XY as example. So this XY, what we are writing, this is nothing but genotype. Phenotype is, you know, that XY is nothing but male, right? So this is a physical expression, that is a male. We are saying XY means is equal to male. So this male, what we are saying, is a phenotype. This XY, what we are writing, is a genotype. So here what is they are saying is both alleles contribute to, for example, this gene is nothing but allele, right? So this both alleles contribute to phenotype of heterozygote. This is heterozygote. What is homozygote? XS, XX. If you take, it is homozygous, like they are same. XY is heterozygote. So this heterozygote is representing only one phenotype. But here They're saying both alleles contribute. Here you can take ABO blood groups as example, especially AB blood group. In AB blood group, you can take A and B. Both are contributing to the phenotype of a blood group. Whereas normally, if you see uh, s- capital X, small x. Capital X is dominant and small x is recessive. Always dominant one will express the phenotype. But here in this case in codominance, that's why the name codominance. Both are expressing the phenotype A and B blood groups. Now coming to variable expressibility. In the name itself you can see there is a variable expression. That is the patients with same genotype have variable phenotypes. For example, they have XX, okay, as a genotype, but they show many different phenotypes. Here the example is neurofibromatosis 1 disease. In this you have the same disease, but they show different, sometime it may affect eye, sometime it may affect skin, so they are showing different phenotypes. Next, coming to incomplete penetrance. For this, the example is BRCA1. You know, this gene is mutated mostly in female breast and ovarian cancer. Actually, this is a germline mutation. So, all the patients who have BRCA1 mutations running in their family, not necessarily they should get the cancer. Even though some girl have BRCA mutation, but she may not have cancer, right? So that is a incomplete penetrance, means not always if we have that kind of mutation we may get that cancer, etc. It's wrong, okay? That is incomplete penetrance. Next, coming to pleiotropy. Pleiotropy is that one gene may contribute to multiple phenotypic effects. Here, for example, you can take phenylketonuria. This is a problem in which it is a one gene only which is leading to multiple phenotype, multiple problems like phenylketonuria. Here it is leading to light skin, it is leading to intellectual disability and also musty body odor. For pleiotropy, you have other examples like Marfan syndrome, where limbs, eyes, blood vessels are affected. In cystic fibrosis, both lungs and pancreas are affected. In osteogenesis imperfecta, bones, eyes, and hearing are affected. Now coming to anticipation. Anticipation is like, you can take example of Huntington disease, which is a trinucleotide repeat disorder. Actually, here the gene CAG it repeats like this. That is known as trinucleotide repeat. Actually, anticipation means, for example, someone is having Huntington disease in their family. So one person got Huntington disease at 50 years. The other one, the next generation one will get it at 40 years, and again next generation will get it at 30 years. So like that you can see increased severity, earlier onset of disease in succeeding generations. In succeeding generations you may get the disease even in 30 years. So like that is called as anticipation. Next, coming to loss of heterozygosity. Heterozygosity is like, you need to understand two hit hypothesis here. The examples are retinoblastoma, Lynch syndrome, Leaf-Frommeni syndrome. Actually, here the problem is uh, retinoblastoma gene. Actually, this is a familial problem where from the birth only the child have one of her gene is already affected from the birth itself, but she will not get retinoblastoma directly. If her another gene is also affected because of some, I mean, environmental causes or other causes, then only she'll get retinoblastoma. So the two genes should be affected to get some particular disease. So that is called as two hit hypothesis. So that is also meaning of loss of heterozygosity. Only then you can get the that kind of cancer or disease. Even in Lynch syndrome, Leaf-Frommeni syndrome, the same type, two hit hypothesis is required. Means the cancer or the gene should be hit by two sides, then only can get cancer. Next, coming to epistasis. Epistasis is the allele of one gene affects the phenotype expression of allele in another gene. Even though they are different alleles and different genes, but it has a capacity to control the other allele on another gene. This is seen in albinism and alopecia. Next, coming to aneuploidy. Aneuploidy is a condition where you get abnormal chromosome number. So you can see in Down syndrome, actually you should have only two chromosomes, right? Pair of chromosomes. But here in Down syndrome, you have three trisomy 21 means you're having three chromosomes on 21st chromosome number. And Turner syndrome, you have monosomy, like 45XO. Here 0 represents there is no one more x. Actually in a female she should have 2 x, but in Turner syndrome she's having only 1 x, that is monosomy. So either there may be extra like in Down syndrome, or there may be less like in Turner syndrome. This is example for aneuploidy. Now let us continue other genetic terms, like you can see dominant negative mutation. Here the example is, you can take P53. With the example we can understand this. P53 is like a mutated, for example P53 is mutated, which is a tumor suppressor gene, right? So as it is mutated, it will produce a protein, and that protein will bind to the DNA, and this will block the normal or natural type of P53 from binding to the promoter, so that now it will not bind to the promoter, means the DNA will not undergo replication, or it will stop that process. So like this, one affected gene, you can see here, a heterozygote produces non-function altered protein, and that prevent the normal gene product from functioning. So a mutated gene is affecting the normal gene, that is the problem here. Dominant negative mutation, one is undergoing mutation, but it is also affecting the normal protein, and it is inhibiting the process normal functioning. Next, coming to linkage disequilibrium. To understand linkage disequilibrium, you need to know thing, that is, can see here independent assortment. Independent assortment is nothing but, you have two chromosomes like this. If the chromosomes are very near to each other, then there's less chance of crossover or recombination. But if the chromosomes are far away, then there is a chance of recombination. So in linkage disequilibrium, what happens is the chromosomes are having the tendency to be close to each other. So as they're close to each other, there is less crossover, and we can measure this type of disequilibrium in population but not in families. So different population will have different degree of linkage disequilibrium. Now coming to mosaicism. Mosaicism is genetically distinct cell lines are present in same individual. For example, you have one set of genes, they should form only specifically this kind of genes normally, but in mosaicism you will form half of this genes and half of this genes. So it is genetically distinct, some distinct types of cell lines are produced in some individuals. This is divided into somatic mosaicism and germline mosaicism. So this problem can occur in somatic means after fertilization, when the child is born, if this undergoes, then this will affect only that particular child, it will not affect her generations like that. But if it is a germline mutation, that is a mutation occurring in agar sperm, then in the child before fertilization only, she can form this kind of mosaic distinct cells. So this may also pass to the future generations. The example for this mosaicism is McCune-Albright syndrome. Actually here this diagram represents it. It's a problem in GS protein activating mutation, so that will lead to unilateral capsule spots and ragged edges, and you can also see polyostotic fibrous dysplasia. In the name itself you can see polyostotic fibrous dysplasia, means normally the bone should be in the in the bone you should have bone material, but instead of bone you will have fibroblasts and collagen filling it. And also you have one endocrinopathy, that is this child will have precocious puberty. And the same McCune-Albright syndrome, if it is present in a germline, that is before fertilization, then it is lethal, but it is survivable in patients with mosaics. Next, coming to locus heterogeneity and allelic heterogeneity. Here locus means you should remember as location. So this genes which are on multiple location, there are multiple genes, but the disease they produce is same. Whereas in allelic heterogeneity, there are multiple genes, they are forming same disease, but the location is single location. So that is the difference here. In locus, multiple locations; in allelic, single location, but these are same, multiple genes, same disease. You can see mutation at different loci, these are same disease. Here the example is albinism, retinitis pigmentosa. Here actually for these diseases, they have mutations in different locus of the chromosome, but they get the same disease. But in beta thalassemia, cystic fibrosis, what happens is only the single location you should get the mutation, then only you'll get that kind of same disease. So that is allelic heterogeneity. Next, coming to heteroplasmy. Heteroplasmy is where you have both normal gene and also a mutated gene, especially in mitochondrial DNA. So this will retain variable expression in mitochondrial inherited diseases. Example is actually mitochondrial DNA is passed always from the mother to the children. That is important. Mitochondrial DNA should be passed from the mother to the children. Either it may be male or female child. They will get their mitochondrial DNA from the mother. Next, coming to uniparental disomy. Uniparental disomy is, for example, you have a female and you have a male. So now the child which is born is, for example, female, then we'll get one X from the father, one X from the, sorry, this is father, right? This is mother. We'll get one X from the mother, one X from the father. Even in the case of male child also, we'll get one X from the mother and Y from the father. This is normal. But in uniparental disomy, what happens is, here the X which you get, both X, if the female child is born, all X from the mother are directly coming to the female child without involving male child, male father, without father. Directly mother XX are coming to the female girl. This is uniparental disomy. In the case of male also, X and Y, this both are directly coming to the male child. They are not fusing with the mother one. So this is uniparental disomy. This is of two types. That is heterodisomy 1, and that is type 1 and type 2. In type 1 there is an error in meiosis 1, so there is heterozygous forms, like you can see here, meiosis type 1 non-distinction 21 ACD CD heterozygous, two different types of genes are passed, whereas in meiosis 2 non-distinction you can see ACC, two same types of genes are coming into the child. That is isodisomy. It is meiosis 2 error. So this is called uniparental disomy. Example for these diseases are Prader-Willi and Angelman syndrome. Actually, here the problem is in Prader-Willi, the male father only will give all the chromosome to the child in Prader-Willi, whereas in Angelman, mother will give the chromosome to the child. Next, coming to Hardy-Weinberg population genetics. Actually, here you need to learn two equations, that is P plus Q is equal to 1. P square plus 2PQ plus Q square is equal to 1. Let us understand this equation. Here P square represents autosomal homozygous allele. You can see homozygous dominant. P square represents homozygous dominant and 2PQ represent heterozygous. Q square represents homozygous recessive. If you see this, you can understand. Here P square, this one AA, homozygous dominant, because capital A, okay, and Q square represents this small a homozygous recessive, because both small a homozygous recessive, whereas 2PQ, you can see here 2PQ, this is representing capital A and small a, that is heterozygous. Okay, so this, if you understand, that is about Hardy-Weinberg population genetics. You can do different kinds of sons by using this formula, and important rules are: there is no mutation occurring at locus, natural selection is not occurring, this is completely random mating, there is no net migration, there is large population in one. So if a population is in Hardy-Weinberg equilibrium, then the values of P and Q remain constant from generation to generation. Let's continue with imprinting. So first you need to understand what is this definition. So for that you take two genes as example, and one gene, it is of course coming from mother and one from father. Okay, and already in imprinting the problem is the one gene which is coming from either parent is methylated. If it is methylated, it is silenced, and the other gene is having some mutation problem now. So already one gene is mutated, silenced, and the other gene which is present, this is also having mutation, and that is leading to two types of problems. One is Prader-Willi syndrome and one other one is Angelman syndrome. Let us see what is the difference between them. The first one is Prader-Willi. I remember Prader-Willi syndrome as a 15-year-old boy who is having some intellectual disability. As his brain is not working well, he eats more and he became more obese. And even as gonads are hypogonads, hypogonadism, and there's hypotonia, that is as muscles have become weak because of eating lot of junk food. So like that, I remember this Prader-Willi. Why I said like this, you'll understand now. So actually, one more thing, this Prader-Willi is in boy, 15-year-old boy. So why I said boy is because, as I said, in imprinting problem, already one gene is silenced and the other gene is having mutation. So already the gene which is silenced in Prader-Willi is mother gene, the gene which is coming from mother, and the gene which is from the father is having mutation here. That's why I said 15-year-old boy. Okay, so the father gene, the paternal gene is deleted or mutated here, and that gene is PWS. Very simple, Prader-Willi syndrome, PWS gene. Okay, and the signs and symptoms now you see here: hyperphagia, obesity, intellectual disability, hypogonadism, hypotonia. Did you get that? Which I said, 15-year-old boy eating more. Okay, the same thing. And the chromosome involved is of course 15. That's why I said 15-year-old boy. And one more thing, that 25% of this Prader-Willi syndrome can be from uniparental disomy. That is only maternal uniparental. Already I discussed this in genetic terms. Uniparental disomy means both the genes are coming from one parent. Here, if both the genes are coming only from the mother, then there's absence of father, because here the problem is in paternal gene, right? The paternal gene is either mutated in the case of imprinting, or it is totally absent in the case of uniparental disomy, especially maternal uniparental disomy. So they give other syndrome like POP, Prader-Willi, obesity, paternal allele deleted. Now coming to Angelman syndrome. In Angelman syndrome, imprinting, I said already, both genes, one gene is already silent. Here already the father gene, paternal gene is silent, and the mother gene, that is the gene which is coming from the mother, is mutated here. So this one I remember like a 15-year girl, because here also chromosome 15 is affected. Okay, so I remember 15-year girl is having some brain problem, so she's laughing a lot, and as she's laughing she got scissors and she fall away. So scissors and ataxia is fall away. So like that I remember this. And here the gene which is mutated is somewhat different. So as she fall away, I remember she booked a Uber cab. So UBE3A genus mutated here. Okay, and one more thing here, 5% of this case same like parental, especially paternal uniparental disomy, because here the maternal gene is not present, everything is coming from paternal, and that is leading to this Angelman syndrome. So here the mnemonic they gave is mamas. It is maternal allele is deleted, Angelman syndrome. Mood is gone, ataxia, scissors. Mood is nothing but like intellectual disability. Funny thing is inappropriate laughter and Angelman syndrome. Before starting modes of inheritance, we need to know the basic rules. The basic rules are like, you need to understand actually the pedigree flow charts. So here if you see, this round color represents female and this square shape represents male, and in this circle you can see it is filled with some color. If anything is filled with a color, that represents it is disease, it's having disease. So for example, you'll try to understand this. It's a female, male, they got married and they are their children, and in that children you can see one male boy is affected and the side one female girl is affected, and again that male married one girl and they got three children. There you can see one male, one girl is affected and another male is unaffected. So like this, just an example to understand. And one more thing you need to understand is, you can see the words here dominant, recessive, right, in every disease. So in dominant, important thing you need to remember is, in any dominant kind of disease, 50% of their offsprings will be affected. Okay, and any recessive kind of disease, 25% of their offsprings will be affected. So how is that? Everything we'll understand now. Okay, first the disease is autosomal dominant. I'll also make you understand how to, just by seeing the pedigree chart, you should be able to say what kind of disease is. Even that we'll learn. So first is autosomal dominant. We'll start with this matching. You can see here one of the parent is recessive, homozygous recessive. One of the parent is heterozygous. So this children, their children are 50% affected. You can see this capital A, capital A. This red boxes represent 50% of the children are affected because it is autosomal dominant according to our rule we said, right? Next, how to determine this flowchart. Important tricks for autosomal dominant. The trick is male to male transmission is seen only in autosomal dominant. That is, you can see here the male who is affected and his son is male and he also got affected. So this male to male transmission is seen only in autosomal dominant. You can see anywhere else. In this, no. In this, no. It is indirect, right? This male and this a female affected, but male to male transmission, no. Here also can see no male to male transmission. Here also no male to male. So only male to male transmission is seen in autosomal dominant. Very important. Autosomal dominant, you can see clearly male to male transmission. That is a key point to find the pedigree chart. And next there is a song to remember the diseases which are related to autosomal dominant. It's like a children rhyme. Von ALS, RPMEN, Tubes Spears in Huntington, Marfan Ehlers-Danlos, NF1 and NF2, don't fab too much, autosomal dominant they are. So this is a song where you can remember all the diseases. It's like a rhyme where you can remember all the diseases related to autosomal dominant. That is von Hippel-Lindau, von Willebrand disease, ALS disease, retinoblastoma, multiple endocrine neoplasia, tuberous sclerosis, spherocytosis, Huntington disease, Marfan syndrome, Ehlers-Danlos syndrome, neurofibromatosis 1 and 2, familial adenomatous polyposis. So all this, actually if you remember them like that, it's very hard. So it's like a rhyme. Von ALS, RBMEN, tubes and spears in Huntington, Marfan Ehlers-Danlos, NF1 and NF2, don't fab too much, autosomal dominant they are. Okay, so this is some rhyme for that. Now according to rule, autosomal recessive means 25% of the children are affected. You can see here 25%. Actually your two parents are heterozygotes. You can see capital A small a, capital A small a, and 25% of the children are affected. And how to see autosomal recessive pedigree easily? Only one child is affected. That is a key for autosomal recessive. Only one will be affected. So okay, this is autosomal recessive, and the song for this is happy birthday song. Alpha beta PKU ferrum copper benzow hartnum Landsman fanconi, autosomal recessive as it's true. So it's like a happy birthday song to remember the diseases. It is alpha for alpha 1 antitrypsin deficiency, beta for beta thalassemia, PKU for phenylketonuria, ferrum for hemochromatosis, copper for Wilson's disease, Burns-Sound syndrome, Hartnum, Glanzmann, Fanconi, autosomal recessive. So very important to remember this song so that it's very easy to remember the diseases. Next, coming to X-linked recessive. Again, recessive means 25% of the children are affected. Very simple, right? And here in X-linked disorders, you need to remember two things again, two common rules again. Whenever it's X-linked dominant, I remember father as dominant and mother as recessive. Okay, so in X-linked dominant, the disease will spread from the father to her daughter. Okay, opposite. I remember the child is of opposite gender because you have father male, daughter female. Here you can see X-linked recessive, I think mother as recessive, so mother to son. Mother is female, so son is male. Opposite gender, a daughter or son will be there. Okay, so your father to daughter, your mother to son. That is a general rule again. And you know already recessive 25%, and it's X-linked. Okay, X-linked recessive. And here also we have another clue to find out this chart. Again, that's it. Mother to son. You can see here, mother is affected and it is passed to the son. You can see from mother, son got disease. Okay, that is a clue here. And the song for this is Hunter's Name, Les Fabry, Short, Menki, Waspen, G6PD, Come Up Bruton, Dochikai, A and B, X-Link. Don't forget diabetes, insipidance, and Turner's. So actually here the diseases are Huntington disease, Lesch-Nyhan syndrome, Fabry's disease, Menkes disease, WASP disease, G6PD disease, Bruton's disease, Duchenne's muscular dystrophy, A and B is hemophilia A and B. These are excellent. Even diabetes insipidus and Turner syndrome. So that is excellent recessive. So as I said already, dominant means from father to daughter. You can see the pedigree from father to daughter is getting the disease. And again, dominant means 50% of the children get affected. Luckily here we don't have any song, only two examples. Fragile X, very simple to remember. You can say X here, okay? Fragile X and Alport syndrome, only two to remember. Also hypophosphatemic rickets, okay? Hypophosphatemic rickets, Fragile X, Alport, example of X-linked dominant. Finally, coming to mitochondrial inheritance. So, mitochondrial inheritance is already discussed that from mother only the disease will go to all children. So, very simple. Here in pedigree also can find from the mother only they are getting disease to the children. So, that is very simple. You may get doubt now, like in X-linked recessive also from the mother you're getting to children, but in X-linked recessive from the mother first it will go only to the son. But here in mitochondrial even female is affected, okay? Like that you can differentiate this mitochondrial inheritance from X-linked recessive, okay? So here the examples are mitochondrial myopathies, labor hereditary optic neuropathy. These are important to remember.